Thanks, Guy, and hello, everyone. So I'm a genetic counselor by training, actually. I'm out at Geisinger Health System out in central Pennsylvania, and I'm going to walk you through today and some of what you need to know about dup15q syndrome, this very complex region of the genome, a little bit of a history of how we got to this today. And I am a PC, definitely not a Mac. How do you So as I said, I'm going to start by just giving an overview of a little bit of the history of the dup15q research and where we've been. I'll start by saying, I can't see my own slides, that a few disclosures. I'm a consultant, and I serve on the board of directors for the National Fragile X Foundation, and I'm a consultant for LabCorp and Ovid Therapeutics. So we are very pleased in recent years to be able to say unabashedly duplication 15q syndrome. In the early days, because some of the clinical features of the condition were subtle, at least the physical features, we really debated whether we could call this a syndrome, whether it was specific enough to call a syndrome. And the last several years we've been able to do this, and just late last year we were able to publish our gene review on dup15q syndrome, put our actual syndromic stamp on this. So in terms of a syndrome, obviously it's a recognizable pattern of physical and behavioral characteristics, and when you have primary characteristics of autism or developmental disability, that overlaps with so many other things. I think that's where the question came up, is this truly a recognizable syndrome? But there are other features as well. The other thing about dup15 is because most of the time, 80% of the time, it involves an entire extra chromosome, this was a condition that was able to be picked up in early days on karyotyping in cytogenetics. So it was known about for many, many decades, whereas a lot of copy number variants are only being identified now that we have microarray analyses. This one was known way, way back, decades ago. And it's had many different names over the years. So many decades ago, it was called an inverted dup15q, isodicentric 15q, which we still use today. Some reports back in the day called it a partial trisomy 15, sometimes referred to as tetrasomy 15q, interstitial dup15, etc. So this can be very confusing, as you can imagine, for families, and one of our first challenges in trying to organize enough families to think about research and support was to gather families who were all being given these different names for the condition and have them realize, yes, we all have the same thing. And the features include infantile hypotonia. There are some subtle facial differences, and the parents will be speaking and hopefully showing some videos as well, and you'll see how subtle they are. These are beautiful kids, but when you go to a conference like we just did and you see many together in the same room, many of them look like they could be siblings. Intellectual disability of varying degree. Epilepsy is certainly a feature for and a significant concern for at least half. And autism spectrum disorder in a majority. There's a very small minority that have SUDEP, or sudden unexplained death in epilepsy. And this syndrome is related specifically to duplication of the Prader-Willi-Angelman critical region on the maternal copy of 15. We now know through the work of Larry Ryder's group in Tennessee and others that there is a paternal duplication as well. But really, when we're talking about the syndrome, we're really focusing on the maternal copy. So just to step back and look at the region, the deletion of the region, as most of you well know, can be seen in two different syndromes, Prader-Willi syndrome and Angelman syndrome. And for Prader-Willi, this was first clinically described in 1956. It was a clinical diagnosis, and there was, again, a syndromic pattern, and there were three primary features, the three H's: hypotonia, which overlaps, of course, with our dupes, hypogonadism, or underdevelopment of the secondary sex characteristics, and hyperphagia, a very strong motivation to eat and compulsion to eat and overeat. And typically in Prader-Willi, there's mild intellectual disability, and Prader-Willi syndrome occurs with a deletion on the paternally inherited chromosome. Angelmans syndrome being the converse with a maternal deletion, and it was first described a few years later after Prader-Willi syndrome, and it's really quite different. The features involve much more significant intellectual disability, characteristic behavioral pattern and facial appearance, frequent unprovoked laughter, and often seizures. Now, what's interesting in DUP15Q syndrome is that in many children who have DUP15Q, when you start talking to the parents, they'll sometimes describe frequent unprovoked laughter. Sometimes they'll describe, a very strong appetite and wanting to food forage. And it's kind of interesting to see some more manifestations of the deletion syndromes, but in a milder form. And they don't really characterize all people with the duplication, but it's something certainly that we hear. So back in the 80s, there started to be many reports about supernumerary marker chromosomes that you could find on karyotyping, even really squatty little chromosomes that weren't stretched out. You couldn't see many bands, but you saw this whole extra chromosome. And these supernumerary markers could come from a variety of different origins, but the most frequent one involved chromosome 15. And so sometimes it was very puzzling because you could see these supernumerary marker chromosomes that seemed to be derived from 15, but they weren't really associated with any particular phenotype. It seemed like they were running in families. And then in other individuals, you saw a very significant phenotype, which we now know to be DUP15Q syndrome, and they were typically de novo. But with advances in chromosomal staining techniques, it became clear that many of these were 15, and we started to really be able to focus, okay, what is it about this group of individuals who have phenotypic effects from having this supernumerary marker? But apart from ID, it was not thought to have a very specific clinical phenotype. Again, the idea of a syndrome. Even well into the 2000s, we still weren't calling this a syndrome. So in 1981, Prader-Willi was linked to the region of chromosome 15q, the 15q11-13 region, and in 1987, these same deletions were reported in Angelmans syndrome, and this created all sorts of consternation and wringing of hands. How can you have two such distinct clinical syndromes, and they're both associated with a deletion? And then by 1989, it was clear that this was a region of the genome affected by imprinting, so that you had maternal and paternal deletions resulting in different characteristics. But then it was in the 1990s that it became clear that these supernumerary markers, these extra copies of chromosome 15, were maternally inherited. And we started to notice, as families came together, and with the origins of this organization, DUP15Q Alliance, in the mid-90s, that, there was a subtle but recognizable clinical phenotype as well. So again, as noted before, there are key features that are involved in DUP15Q syndrome. And I do want to point out two of the features that really should be of great interest to researchers, just because there's a lot of research in this area now. Certainly the epilepsy phenotype is really important. We know lots of studies are going on looking at the genetic basis for some of the epilepsies, and we do know some of the genes in there, particularly the GABA receptor genes that are involved in this region play a role in the epilepsy phenotype. So that's a really important part of the phenotype for research, because there are a lot of other collaborators and scientists who don't know anything about DUP15 but are interested in this area. Autism spectrum, of course. Many people are working on the genetic underpinnings of autism spectrum disorder. As it turns out, in clinical samples in the laboratory sent for a indication of autism spectrum disorder, the most common copy number variant they find is this one, the DUP15Q. So this is a big player. We know that there are genes involved that cause changes in neuronal function that result in the behaviors that we see that we call autism. So if we learn about DUP15Q, we may learn quite a bit about autism in general. So just show a few pictures of kids, and there's quite a bit of variability. The clinical or physical phenotype, the facial features are not especially distinct, and these children and adults do not really look unusual out in the general population versus anyone else. But for those of us who are deeply involved in meeting hundreds of people and families, we can see the similarities. We truly are a family of, oops, I think I something. We are a family of and a community, and brought together also because at every conference you hear parents say, oh my gosh, that child looks so like my child when he was young, etc. So there are some similarities. Again, one couldn't point to it and say it's incredibly obvious dysmorphic features, but we see just some similarities, sort of a low nasal bridge, sometimes hypertelorism or white space between the eyes. Really, it's more of a gestalt than anything else, and when you see it, you say, yeah, now I think we're confident enough at our centers and the other clinics for DUP15 that when we see a kid come in, we can sometimes say, boy, that child, we definitely should be zeroing in on DUP15. So in terms of history of the DUP15Q Alliance, so the original organization that led to the DUP15Q Alliance was known as IDEAS, and it was actually a parent who had a son with DUP15Q, an isodicentric supernumerary chromosome, who was at our center outside of Philadelphia, a center called Elwyn. And she and I were combing the literature to see, and looking in parent magazines, like Exceptional Parent Magazine, to see, are there other parents out there who have kids with this after a child was diagnosed? We didn't have the internet quite yet then, so everything we were doing was sending letters and calling and trying to get a fledgling group of families together. Boy, it's so much easier now to form a group. But we started this group called IDEAS, Inverted Duplication Exchange Advocacy and Support. We had 13 families when we started there. We had a newsletter that we sent around. And then in 1997, the cytogenetics or laboratory nomenclature changed somewhat, and they were referring to this as an isodicentric rather than inverted duplication 15. Thank goodness the letters still fit with IDEAS, because we had just printed new letterhead and we didn't have a whole lot of money, and I was like, oh, dodged a bullet there. So that became the Isodicentric 15 Exchange Advocacy and Support. In 2001 and 2003, the first conference was held in Philadelphia. That's when our group and some of our original founding folks are still involved, the scientists and the clinicians. And we started to think about this. Could we be so bold as to call this a syndrome? In 2004, IDEAS was incorporated. And then in 2011, the name was changed to DUP15Q Alliance. And then in 2012, we had our first sort of dedicated scientific meeting, although scientifically we used to get together at other meetings and do what we can. And actually, this is, if I'm not mistaken, the first time we've had a full 2-day scientific meeting. I might be mistaken on that, but it really has grown from a little, let's have some coffee at an IMFAR meeting, to let's have a dedicated couple of days just on the science. So as Vanessa mentioned, there's over 1,400 families now internationally, which is pretty amazing. There's a professional advisory board, and there are some major research collaborations and initiatives that the DUP15Q Alliance has been with. And what I can say about this has grown from a family organization that's interested in this particular condition, and it's been really a collaborative effort with families and clinicians and scientists, which is great. So very quickly, the prevalence of DUP15Q syndrome in the general population has been thought of to be possibly as high as 1 in 5,000. That's from a paper in 2014, possibly around 1 in 10,000. It's hard to know what to quote, and that's not just for DUP15. Any of these syndromes that quote you a prevalence figure, a lot of times you have to take that with a grain of salt. We really don't quite know, but this is certainly, among rare disorders, one of the more common. We know that these duplications are the most common CNV detected in clinical samples with ASD, and about 1 in every 500 clinical samples that are sent for evaluation of developmental delay or intellectual disability or autism spectrum disorder ends up having DUP15. So it's estimated that about 1 to 3% of all autism is due to this particular copy number variant. And then just to show you a few graphics about what we're talking about with the DUP. So this region, in addition to having variations on how you can get DUP15Q syndrome, also is one of the regions affected by imprinting. So maternal versus paternal is important when we're thinking about this. So if we think about maternal and paternal 15s, the regions and the variables we're talking about is when we have somebody with this syndrome, is it supernumerary? Do they have a whole extra chromosome derivative? Or is the extra genetic material embedded within one of the existing 15. So supernumerary versus interstitial. We have to think about de novo versus familial. The supernumeraries are virtually always de novo. We don't know of cases where they're not de novo, but some of the interstitials are familial. We want to know what's the copy number. Some people have 2 extra copies of the region. Some have a combination of 2 copies of some parts and 1 extra copy of the others. And there are even more than that. There are people with hexasomy and other variations. We want to know about breakpoints. This is a recurrent CNV area. So, there's 5 different breakpoints that we talk about in the region, and you can have a number of different combinations in different people. And who's the parent of origin? Is it maternal or is it paternally derived? And when we're talking about the syndrome, again, we're talking about maternally derived duplications. So for the supernumerary chromosome 15q duplications, we also call these isodicentrics, although technically not every single one is technically an isodicentric if you want to get down to it, but these are maternally derived isodicentric derivative chromosomes, and this accounts for 80% of dup15q syndrome. This has always been a little bit of a puzzle because if you think about it as the opposite of Prader-Willi-Angelman with the interstitial deletion in those conditions, you would think you would see many more interstitial duplications than we do. Interstitial duplications are relatively rare. They only make up about 20% as far as we know of these duplications. The rest are all these isodicentrics. There's variability in breakpoints and copy number, clinical variability as well, and not always predicted by size. We do know that as a group, individuals who have an interstitial duplication tend to be somewhat higher functioning, have fewer effects than those with the isodicentric. But even within the isodicentric, you can have 2 kids with exact same extra tetrasomy of 15q, and they really are quite different in terms of their functioning. So there's really not a way to predict very accurately based on copy number. Mosaicism's been observed, and again, for these isodicentrics, there have been no reported familial cases. So if we look at the region, and here's chromosome 15 lying on its side here, and we pull out this portion, what we're talking about here in this syndrome is really duplication of the Prader-Willi-Angelman critical region. You can see across there that there are 5 different breakpoints, 1 through 5. And really it's between breakpoints 2 and 3 that we have the key genes that are involved when deleted result in Prader-Willi or Angelman syndromes. And for the duplication syndrome, it's really duplication of that region. Now what we know is that there are other 15q duplications, for example, between breakpoint 1, breakpoint 2, or on the other end, breakpoint 4, breakpoint 5, that don't involve the Prader-Willi-Angelman critical region. And they may have neurodevelopmental symptoms, but they don't have this syndrome. So the duplication is really defined by including the Prader-Willi-Angelman critical region. And so what does this isodicentric look like? Well, if we lopped off the top of one of the maternal 15s and duplicated it and flipped it over and then reattached it, that's what it looks like. It's kind of like a candy with 2 little satellites at the end most of the time, so it's a dicentric chromosome. And so in terms of copy number, a child may have 2 normal 15s, 1 from mom, 1 from dad, and then they have this derivative that itself has 2 copies of the P arm, but more importantly, the Q arm involving the PWACR. So the person has tetrasomy 4 copies of that region. Carolyn Shannon is responsible for a lot of these really nice graphics here, but there are 2 very common types of IDICs. Sometimes they're symmetrical, so you'll have the same amount of material on each duplicated side, but sometimes they're asymmetrical. So 1/2 of the isodicentric may be somewhat larger than the other one, so you might get 3 copies of total of one region and 4 copies of the rest. And again, we can't predict prognosis based on those copy numbers. So again, this is a slide that shows if you're looking here at the region that's in breakpoint 3 to breakpoint 3, one of the more common isodicentrics, what you have is a symmetrical duplication of this region here. For the second one, which is somewhat larger, you have breakpoint 3 to breakpoint 3, but then extends up to 4. And so you have 2 sides with that. But then in addition, one of those parts of the derivative has part all the way through breakpoint 5. So you end up with tetrasomy for some parts and trisomy for other parts. Again, it's very variable, but these are important things maybe down the line, since there are gene dosage effects that might help us to understand some variability in the phenotype. The other type of DUP15Q syndrome has to do with interstitial duplications. This accounts for about 20%. 80% that we know of are due to that whole extra chromosome. They have 47 chromosomes. For these individuals, they have 46 chromosomes, and the extra material is embedded into one of their existing 15s. Again, we're talking about maternally derived here. There are some paternally derived interstitials, but we don't consider that part of the syndrome, really because of the imprinting effects. And so the mechanism is that we're interested in this particular syndrome is related to the maternal imprint. So there's variability again in breakpoints and copy number. The clinical phenotype is often less severe than in the isodicentric 15q because there's a dosage effect, a lot of times it's triplication, but you can't always swear to it. Sometimes, you know, I can't tell, and I know hundreds of people with this condition now, I really can't tell meeting somebody right up front. I wouldn't venture a guess always to say, because I've met people with interstitials who are have a lot of challenges, and then I've met people with isodicentrics who are very verbal and you would think have fewer challenges. So it's really hard to predict, but as a group we can say that interstitials are less involved. It's de novo in 85% when we're talking about maternal interstitials, but we always want to check. So as a genetic counselor, somebody comes in, they have an interstitial DUP15Q, first thing I want to do is parental studies. Is it coming from one of the parents? And then we may want to know if there's a difference in phenotype, you know, do some additional studies and see where's the parent of origin. Is it paternal or is it maternal? There is a paternal interstitial DUP15Q. It's not the syndrome, but it has a much wider range, I think, of developmental and psychiatric effects, and even individuals who have very few clinical effects. There are some parasomnias involved, and it's got a characteristic EEG pattern that's been described. Can also be seen in maternal DUP15Q syndrome. And that disorder, the paternal interstitial, is often familial. So I think it's important, and maybe it's just because I'm a genetic counselor, to go through this stuff, especially for researchers who might be thinking about clinical research. Because what I've seen, not necessarily in DUP15, but now as we have other copy number variants, sometimes clinical researchers are lumping deletions and duplications, for example, of certain CNVs together and sort of trying to phenotype them. And they may be, deletions and duplications are very different disorders. Well, within this is even more complicated because is it maternal, is it paternal, is it interstitial, is it isodicentric? So some lumping is possible, but you want to be aware when you're lumping and be able to do your analyses and sort out the various types, because there really could be differences based on dosage and copy number and parent of origin. So the interstitials, if you knocked out, if you imagine that this is the region of interest with the Prader-Willi-Angel critical region, and it's embedded into one of the existing 15s, and then so here's the maternal and paternal copies. I think, yeah. And again, I won't go through these slides, but again, we're talking about this region that's involving the Prader-Willi-Angelman region. Even within the interstitials, you may have very large sections that extend from breakpoint 1 through breakpoint 5 even, and they're embedded within a chromosome. So it's not always just restricted to that region. So it's a very complicated region of the genome, and here's from our recent publication in Gene Reviews, just some of the comparison clinically, and I think we're in our infancy in the clinical description of this, and I'm really excited that, you know, at UCLA kind of leading the pack, we're finally getting some good, deep clinical phenotyping, because we've had a lot of genomic phenotyping, if you will, or genotyping, but we haven't had all that much clinical phenotyping. But there are some differences, and you can see differences in severity and prevalence of various disorders. There are other duplications, something to be aware of. As I mentioned before, they're kind of outside on either side of the core region involving the Prader-Willi-Angelman critical region. So the 15q11.2 duplication, breakpoints 1 to 2, doesn't include this region, but may have a variety of developmental effects. It's still considered a variant of uncertain significance. On the other side, there's the 15q13.2 to 13.3 duplication. We know that the deletion of that region causes a wide variety of neurodevelopmental effects, and the duplication really does seem to confer some risk for effects as well. But again, the mechanisms of those do not necessarily involve the Prader-Willi-Angelman region. So when you're thinking about research, you have to think about all these factors. Are you in the right region? Are we talking about the right duplication? Are you talking about the maternal one? And then you've landed on the syndrome. Just a slide from a couple years ago, actually, about the makeup of from the lab reports that we reviewed from the Alliance. You can see that the majority at that time, when we had 186 lab reports reviewed, majority had isodicentric extra chromosomes. We had 16% who had interstitial chromosomes, and then we also had some people in the Alliance at that time who had the flanking regions or those edge duplications. And really, that's a little bit of a disservice. They have different conditions, and I think the Alliance has made a great effort in trying to direct them to resources that are more appropriate for them. So again, there's many things you can have. You can have something, whether it's a supernumerary or an interstitial involving the PWACR, or you can have kind of these flanking edge duplications that are either larger or smaller and either distal or proximal to our region of interest. So as we head off into these 2 days of hearing about a lot of the research that's going on, here are some thoughts for directions for research, some of which we're already involved in. Clinical phenotyping, this is a great need for this syndrome. This is an unusual syndrome in that kind of the laboratory finding was well documented long before the clinical features. In many other conditions like Prader-Willi and Angelman, they figured out the clinical syndrome first, and then they figured out what the genetics were. This is kind of the opposite. We knew the genetics, but we were just getting a handle on the phenotype. So what's the neurodevelopmental characterization? What's the trajectory, long-term span, and what's the natural history, if you will, over the lifespan of what this looks like? What are the psychiatric comorbidities that we see? Epilepsy is really an important aspect of this, and one really that's worth looking into, because epilepsy is real hot right now in terms of targeted treatments for epilepsy based on some of the genomic underpinnings in other disorders. The unexpected death issue with SUDEP, which actually is found in a number of other conditions that involve severe intellectual disability and epilepsy. So this is not unique to DUP15. Also other medical issues. You'll hear over the next few days about animal models. This is really important. These are the first steps in really understanding pathophysiology and moving toward targeted treatments. And what are the shared neurobiological pathways with other etiologies of autism, ID, and epilepsy? And this is really important. Conditions like Fragile X syndrome are light years beyond, in many ways, many other conditions. They've been at it longer. They have a single gene. They have a simpler mechanism. But what can we learn about Fragile X, or what can we learn about tuberous sclerosis? How do we compare and contrast and figure out where are the shared underpinnings so that we can piggyback some of our research on what other researchers are doing? This is very exciting now that a lot of neurodevelopmental researchers really are talking to people with other syndromes. In the past, you know, I'm a genetic counselor. I cross a lot of syndromes, but I'd have to remember, oh, today I'm talking about 22q11.2 deletion. Entirely different set of researchers than DUP15, entirely different set of researchers than Fragile X. That's a very inefficient way of doing things. So really, collaborations are encouraged. And then, of course, leading toward the mission of the DUP15Q Alliance, things on the foremost of the minds of the families living with this and the individuals. Really, can we come to some targeted pharmacological and behavioral interventions that really improve quality of life?
He's the boss. Okay, bye.